Hello, everyone. My name is Shivani Babu. I'm a junior from Auburn High School, and I'm very excited to talk to you today about my project entitled The Attenuation of Adipocyte Hypertrophy in a Palmitic Acid-Induced 3T3 cell model through Lupeol treatment. I'd like to start off today by talking to you a little bit about what obesity is, which is the enlargement of adipose tissue that is caused when more calories are intaked than they are released. And this can have a variety of negative health detriments, including but not limited to diabetes and cancers. And this is a major problem worldwide. And if we just focus on the United States, 32% of adults are classified as obese. The purpose of my study was to find a treatment that would hopefully reduce the effects of obesity, or specifically the metabolically unhealthy form and irreversible form of obesity, which is adipocyte hypertrophy. I planned on using Lupeol. Lupeol is a phytochemical found in fruits and vegetables and has been previously shown to have anti-inflammatory effects. The purpose of my study was to use this to determine whether this could be used to attenuate adipocyte hypertrophy. So I'd like to go through with you today my design and methodology. So I had four main groups. I first had the control, which was the mature adipocyte. I had the control that was treated with Lupeol. I had the hypertrophied adipocyte, and I had the hypertrophied adipocyte that was treated with Lupeol. So all four cell groups for the first 10 days were treated with an insulin medium in order for these pre-adipocyte cells to turn into adipocytes. After that, groups 1 and 2 for the next 2 days were treated with a treatment. The control was treated with a DMSO treatment, and this was done because Lupeol is dissolved in DMSO, and the Lupeol control was treated with Lupeol for 48 hours. And for groups 3 and 4, they, for 6 more days after the insulin medium, were treated with palmitic acid in order for these adipocytes that were formed in day 10 to become hypertrophied adipocytes. After that, they were also treated with DMSO treatment and Lupeol treatment, respectively. After this, the cells were collected and analyzed using oil redo staining and Western blot analysis. So my study had 5 main goals, with the main aim of the study being to evaluate the impact of Lupeol and whether this could be used as a treatment against adipocyte hypertrophy. My first sub-aim was to determine whether these pre-adipocytes in day 0 had turned into adipocytes. And this was done through 2 methods. First, by staining these adipocytes and pre-adipocytes with an oil redo staining that binds specifically to lipids and determining whether there was more or less stain found. And number 2, by doing a Western blot analysis and looking for a specific protein called FABP4, which is a protein that binds to fatty acids. And if there's more, there is more lipid content within the adipocyte. And as seen in both, there was a much higher lipid content and much larger expression of FABP4 in the adipocytes when compared to the pre-adipocytes in the beginning, thereby confirming that the adipocytes had formed. The second sub-aim was to verify the development of hypertrophy for groups 3 and 4 by checking the area of lipid droplets. So as you can see here in figure 2, the adipocytes have much smaller lipid droplets in comparison to the hypertrophied adipocytes. Not only can we tell this visually, but also statistically, the adipocytes had a much larger amount of smaller lipid droplets in comparison to the hypertrophied adipocytes, meaning that the hypertrophied adipocytes had a much larger amount of larger lipid droplets. The third sub-aim was to verify the best Lupeol dosage that would have the highest impact on fatty acid binding protein 4 expression. Like I mentioned earlier, if there's less expression, that means that the lipid content is decreased. And as shown in my Western blot analysis, the best amount was 60 micromolars, which was the dosage that was used here in the cell culture after determining that it was the best dosage. My fourth sub-aim was, after the cell culture and after the cells were collected, was to determine how the lupeol was affecting these hypertrophied adipocytes through lipid accumulation and oil redo staining, which I talked about in sub-aim 1. As you can see here, the control and hypertrophied adipocytes have much more red staining here, as you can see, in comparison to the control treated with lupeol and the hypertrophied adipocytes treated with lupeol, showing that the lipid content was decreased after the lupeol treatment. To double verify this effect, I looked at the FABP4 expression that I mentioned earlier, as well as adiponectin and PPAR gamma expression to determine whether the lupeol had effect on the expression of these proteins and markers. All three of these usually are expressed more when there is a higher lipid content. And as you can see in figures 5, 6, and 7, the hypertrophied adipocytes were shown to have a much significantly higher expression of these proteins in comparison to the hypertrophied adipocytes treated with lupeol. In conclusion, this study confirmed my hypothesis that lupeol treatment would result in a lower lipid content and expression of these adipogenic proteins and markers. This now can be applied in the outer world. Now we can look forward to hopefully utilizing more lupeol in our diet, or perhaps even creating a lupeol supplement that could be used to attenuate the effects of adipocyte hypertrophy. Thank you to Dr. Judd for helping me throughout this project, and thank you for listening.